Hello, and thank you for joining us today. My name is Janie Sweet, and I'm president of the American Technion Society Board of Directors. I fell in love with the Technion and the professors and the students on my first visit to campus in 1989. Technion's impact on science, biotechnology, and medicine for the benefit of Israel and humankind touched my heart and sparked my involvement, which has continued now for more than 30 years. It's my hope that what you hear today on this webinar will touch you as well, and I invite you to join with us. As we all know, it takes a village of scientists to tackle the increasingly complex health and medical issues of our time. Advancing the standard of healthcare in the 21st century will require the same multidisciplinary efforts that have led Technion to breakthroughs in oncology, neurological diseases, and COVID-19. The Technion Human Health Initiative, shorthanded to THHI, brings together physicians from the Rambam Health Center in Haifa, Technion researchers from several faculties, and industry partners in the pharmaceutical and biomedical sectors under one roof. This comprehensive collaboration hopes to support advanced human health research and to help transform discoveries into applications and products for people worldwide. The THHI will represent the most ambitious endeavor in the history of the Technion and will be unmatched in Israel in terms of its breadth. It will stand among a handful of similar initiatives worldwide taking such an extensive approach to tackling these problems. Our guest speakers today will represent just a small sampling of those involved with the THHI, but their work is wide-reaching in areas including cancer, memory loss, and infertility. I'm delighted to be joined by a few people who will be tackling these issues. Professor Noam Ziv, Director, Technion Human Health Initiative, Professor Philippa Melamed, head of the Molecular Endocrinology and Epigenetics Laboratory in the Faculty of Biology, and advisory board member of the Technion's Human Health Initiative, and also Professor Nabieh Ayoub, a cancer researcher in the Faculty of Biology, as well as Deputy Dean for Graduate Studies. Thanks to all of you for being here today. Just a reminder, we'll have time for questions after all of our speakers speak. Please use the Q&A function on the webinar screen to submit your questions, and we'll get to as many as we possibly can. Now it's my pleasure to introduce Professor Noam Ziv. Professor Ziv is not only the head of the Human Health Initiative, but also the Deputy Senior Vice President for Biomedical Science and Engineering, and is a member of the Ruth and Bruce Rappaport Faculty of Medicine. Noam, we're thrilled to have you with us today. The program is all yours.
Thank you. So thank you everybody for joining us for this event. As said, I currently head the Technion Human Health Initiative, and I'll try to give you a few words about what it's all about. So the mission statement of the THHI is defined as to drive research toward groundbreaking advances for the benefit of human health by creating and fostering opportunities for synergetic interactions among Technion research groups, centers, facilities, and affiliated institutes. Well, that's quite a mouthful, and I'll try to break it down and explain what it actually is all about. So in an ideal world, the THHI would look like this. We would have our many people working in engineering and natural sciences and life sciences and data sciences, working together to address things like cancer, brain disease, aging, and this would result in medical devices, perhaps cell and molecular medical technologies, and perhaps medical data integration. The thing is that Technion, the way it's built is 18 faculties which are dispersed around a rather big campus. And in fact, if you ask many of the people in engineering faculties, for example, they have many interests. So for example, mechanical engineering, they're interested in machines, not necessarily in health. In electronics, people have the whole big high-tech industry, which interests them. And therefore, one of our major challenges is one of intellectual engagement, and I'll also say cultural change. So intellectual engagement means we really aspire to spark interest and engage investigators and students from various disciplines who perhaps are experts in their field, but by introducing problems and challenges from the world of health, human health, they can suddenly say, hey, actually, this is a technology I can actually apply toward this, or this is a very interesting problem, which I think that will interest me as an investigator, and I can contribute to. The matter of culture change is the matter of connecting research across disciplines, faculties, campuses, and affiliated institutions. Because as I said, we're 18 faculties. We have a big hospital and many affiliated ones as well. There are industry all around us, but people don't always know each other and are aware of what is possible by basically getting to know their colleagues. And all of this has to be supported by infrastructure and, most importantly, facilities for joint research. So the THHI, one more thing. I think the motto for the last year has been, rather than telling our investigators what they should be doing, the best way is bringing them together, and the magic is guaranteed to happen. And this is in quote marks because in some article about the THHI, this was attributed to me. In all honesty, I don't remember ever saying it, but if I did, great, because it's actually quite smart. So what do we actually do? So we work in 4 major directions. Our core mission, research and education. In this regard, we're not different from any other faculty, any other university, because these are the core missions of the university. But we've been working very hard on things that are a bit softer. We're trying to fuel people. And perhaps the major word here is community. We're trying to create communities of investigators that will be interested and work together on a shared problem. These are foundations, the community and the infrastructure for actually doing the work. And hopefully, this will lead to discovery through research and the infrastructure. Now, as I said, because the Technion is built as 18 faculties and a management, and there's also the hospitals, and there's the Cancer Institute, and many other entities, we need a way of actually bringing and harmonizing all the work. So at the center of the THHI is the Council for Biomedical Science and Engineering. It's a fancy word, but all it actually means it's a forum where we have representatives from basically almost all the faculties in the Technion. We have representatives of management, and just as important, we have representatives of the many administrative branches that are actually responsible for taking our ideas and actually translating them into action. And we meet every 2 weeks. We make decisions, then everybody goes home and they make sure that these things happen. So at the center of the THHI is this council. Now, as I said, perhaps the most important thing is to create communities. And therefore, in the last year, we've been working very hard on creating many opportunities for people to meet each other, to get to know their neighbors, to brainstorm, to come up with new ideas, and hopefully come up with new research themes that will be kind of Technion-wide. And some of these are kind of emerging. So if we're talking about neuroengineering, healthy aging, which is an institute, and we're hoping to start establishing in the next few months. Applied physics. Physics is a venerable, very well-respected field, but we're now trying to make it more applied toward issues of human health. Medical imaging, we have really amazing people who are working on devices like detectors for X-rays, for machine vision, people who work on MRI, trying to create a community of people who are working on imaging and create the next generation imaging devices for human health. And of course, you probably heard about artificial intelligence. The Technion is pretty strong at it, and we've been working very hard to merge it with medicine. All of this needs a home. And we were very lucky to receive 2 very significant gifts. The first is the André Cohen de la Rue Building for Transformative Biomedical Science and Engineering, which is supposed to be built by the end of 2025. There's a picture here on the screen. Actually, Philippa, which you will meet in a minute, is part of the committee where we're trying to design it so it fits the purpose. The second gift we received somewhere in June is the Larry and Andy Wolf Center for Engineering and Medicine, which will be 2 floors in the Discovery Tower right inside the Rambam Hospital, which will be a joint endeavor of both the Technion and Rambam Hospital, where we hope that we will enable clinicians and engineers to work together and come up very rapidly with real solutions for the real world. And there's a third building where the negotiations are in progress. So this is on kind of the tip of what's going on. Just a few things I'll just mention, but not go into. We're working very hard on new graduate programs that will be campus-wide on human health. We've been working together on recruiting staff investigators on a strategic basis. We've funded 3 Technion-wide research projects. One of them is this idea of closing a loop between machine learning and artificial intelligence and the clinics, where we have people from the Technion collecting data from the hospital, coming up with ideas how to use it, working with clinicians to test it, going back to the design, and finally turning this into actually something that works. And the other issue we've been working very hard on is the matter of infrastructure, which is very costly and very expensive, but something we really been putting effort in, because this is basically the fundamental layer for doing all the research we do. So that's about it. And we've only been around in earnest for maybe 2 years. And so we're just getting started, and we invite you to join us on this journey. So before signing off, just a word about myself. So we're switching gears here for a second. I'm actually a cell biologist or neurobiologist of brain cells. And we've been working on something we call synaptic instability and perhaps forgetting. And just to kind of take all the jargon and make it very simple, we can peel off all the layers of the brain and say that, in essence, this fancy machine we call the brain is a vast network of cells connected by wires, and these wires are connected onto each other by something called a synapse. This is a drawing from Santiago Ramon y Cajal. It was done about 100 years ago. He, by the way, got the Nobel Prize more than a century ago for his pioneering work. And in more modern terms, you can see more or less the same kind of a network of wires and connections using the very sophisticated machines we have now. And this is a picture from Harvard, from the lab of Jack Lichtman. Now, if you look at these connections, these synapses, then schematically they would look like this. You have one cell, you have a second cell, and they connect by some device. It's called the synapse. Why is the synapse so important? Because it's almost dogmatic that the way the brain changes is not by changing the cells themselves or even the wiring, but the connections. So if you want to embed new memories into the brain, you have to do something about the connections. You can make them stronger, you can make them weaker, you can take them out, or you can add new ones. And that's how the brain stores memories. Now, if you look kind of close up on one of these synapses, then you realize that they look kind of simple in the cartoon, but they're really, really sophisticated devices. This is a picture of only one half of the synapse, the transmitter, if you like. And if you look even closer, just this little tiny piece of the synapse, kind of look up close, it looks like this crowded, crazy organization made out of many, many, many parts. These parts are proteins, but for the purpose of the discussion, think of them as parts that make up the synapse. And you see there are hundreds of these different ones. Each one is a very sophisticated machine. But for the moment, just think of them as components or parts of the synapse. Now, synapses can last for years, perhaps lifetimes, but the parts themselves, as we have found, and others, that the parts themselves are continuously replaced. Anywhere between a few days and a few weeks, these parts are turning over. And even worse, the parts themselves, they wander in and out and between synapses. So kind of to give you an illustration, imagine you have 2 cars parked in your garage. You go to sleep, you come in the morning, you discover that the steering wheel for one car is actually now in the second car, and the parking brake of the second car is now part of the first car. This is what the synapses look like. So one can say, given that all these parts of the synapses are completely replaced every few weeks, then how does this affect the stability of these connections and, by extension, the memories they're trying to encode? So our group has been working on this for the last more or less decade. We've been developing automated microscopes to measure synaptic stability over days and weeks. And just kind of to illustrate, this is a movie from our lab where what you actually see here are, these are kind of wires, and these colorful dots, every dot here is actually a synapse. And the reason they're colorful is because they're expressing, our parts here are tagged with like these fluorescent neon-like appendages, which you can measure with a microscope. And just to give you a feel, if you track these over time, you'll see that the amount of parts, the number of parts you find in any one of these synapses is continuously fluctuating. In some cases, like this guy over here, if you look at it carefully, at some point it just disappears. So we're trying to understand how the brain even stores anything with these kind of very unstable devices, and even worse, what happens when you have defects in these parts, like mutations, and most importantly, what happens when we age or we're subject to neurodegenerative diseases. And at the end, does this have anything to do with forgetting? We believe it does, and we're working on this very hard. So thank you for your attention. I would like to introduce my colleague, Philippa Melamed. Philippa, please.
Thank you, Noam. Good evening from Haifa. Good morning to you, or good afternoon, wherever you are around the world. As you've heard, I'm from the Faculty of Biology and also part of the THHI, and particularly interested in the thematic group, which is looking at healthy aging. So our take on aging is that healthy aging actually starts in childhood. And we're particularly focused on reproductive aging and the reproductive lifespan, because when the reproductive lifespan terminates, then we have this rapid onset of age-related diseases. And this is crucial for us now as our lifespan is extending. We find that this post-menopausal period for many women can be as much as 30 or 40 years. Now, we also know that the age of menopause and also the age of puberty onset is variable. It varies a lot between individuals, and it seems to have some kind of flexibility. Also, other reproductive characteristics, our cycling, our reproductive hormones, they also show a lot of variation. And we're really trying to understand what underlies this plasticity in order to utilize the plasticity for the benefit of our own health. So my lab studies reproductive hormones and epigenetics. Hormones are these chemical messengers that are produced in certain places in the body and travel usually in the circulation to other tissues or cells, changes their function, and these hormones really regulate our physiological systems. The epigenetics is a field which describes the way the DNA and our genes are packaged. And this is really crucially important because the packaging of the DNA reflects the way, the levels in which genes are expressed into proteins. So this kind of packaging, which includes DNA methylation, which I'm sure you've heard of, affects the levels at which proteins are being expressed, and it's very sensitive to different signals and different kinds of environments. This means that because of this sensitivity, changing this gene packaging is also a platform where we may be able to manipulate the levels of proteins which are being expressed. What's also important to note is that both the hormone levels and this epigenetic packaging changes through our development as our physiology changes, but also in response to the normal aging process. If we look at the reproductive axis and see what happens at the time of puberty, when it becomes activated, we have a brain hormone called GnRH, which is activated and it switches on the expression of two hormones in the pituitary gland called LH and FSH. The pituitary gland sits just underneath the brain. And this switch on involves the GnRH changing all of this epigenetic packaging. These hormones, LH and FSH, then travel through the circulation and act on the testes and the ovaries. Because this hormone GnRH sits in the brain, it gets a lot of input and feedback on the environment, the internal environment and the external environment, which translates the message of whether or not this is the right time to enter puberty, to enter sexual maturation, and also in times of stress, nutritional or resource depletion, then the reproductive axis may be switched down. And this again can be through changes in the epigenetic packaging of the genes. So we're interested to understand the role of this epigenetic packaging in the normal function of the reproductive axis, and also in the variation of different kinds of reproductive phenotypes in response to the changing environment. We're also interested in the way the hormones that come out of this axis affect the epigenetic packaging and also the healthy aging in a more broad term. So how do we do this? Looking at the reproductive tissues in humans is very difficult because they're inaccessible. What we can do is we can look at human data, large big data, large cohorts. We can look at reproductive phenotypes in different environments. We can sometimes get access to the hormone levels. And we can look at some of these epigenetic markers in, for example, blood cells or in the lining of the cheek, which gives us some indication of, for example, methylation levels, but doesn't necessarily reflect what's happening in the functional tissues. So for this, we do need to use animal models. We do a lot of cell culture, and we use other techniques in the lab to try and understand the mechanisms behind the phenotypes that we see in the human cohort populations. So what we're trying to do is going from observations of human phenotypes to establish the causality, to understand the mechanisms. And when we understand the mechanisms, we can identify potential biomarkers and also find ways of possible intervention to change the phenotypes. And at this point, I'd just like to stress that one of the aims in the healthy aging thematic group that we're trying to establish is to build up new cohorts of human data from our local populations to establish groups of individuals that we can follow in longitudinal studies and watch them as they are aging. We're also trying to expand our animal facilities in order to utilize better models for human aging than some of the current animal models that we're using. And these are areas that we would really appreciate your support if anyone is excited about the research as we are. The other thing we're trying to do in this new initiative is to bring together different groups of experts. For example, people who are really good at the machine learning and the AI to really take advantage of all of this data coming out of the human cohort studies. And on the other side, those people who are really good at drug development, so that once we've understood the mechanisms and how we want to make our interventions, they can help us take our findings to a truly translational endpoint. So what I want to do today is just to talk very briefly about two short studies that we've been carrying out in our lab, which I hope will demonstrate the utility of this kind of approach. And the first one is about the impact of the early life environment on female reproductive longevity. So the reproductive lifespan obviously starts at puberty, and for women it ends at menopause when the number of eggs in the ovary drop below a minimal level. What you can see on the right in the graph in green is the number of eggs that are present in a woman's ovary through her entire lifespan. So the eggs are produced when she's still an embryo, they reach a maximal level, and then they start just dying off. So a woman is actually losing her egg supply throughout her entire lifespan until she reaches the point of menopause. So what dictates the age of menopause is the number of eggs that she starts off with and the rates of loss. And it's the rates of loss that can really vary in response to the environment. So this study we carried out with a group of scientists in the UK who were working on migrants from Bangladesh who were living in either the UK or the Bangladeshis who stayed in their home country in Bangladesh. And they showed that the reproductive lifespan was very different depending on the childhood environment. So the Bangladeshi women who grew up in Bangladesh had a shorter reproductive lifespan with a later puberty onset and an earlier menopause, compared to the Bangladeshi women who grew up in England, either child migrants or second-generation Bangladeshis, who had a longer reproductive lifespan. These Bangladeshi women are not short of nutrition. They don't carry out physical labor. But what correlated with this different reproductive phenotype was a higher incidence of inflammatory disease in response to slightly less sanitary conditions in their environments in which they were growing up. So from here, we put together a mouse model of early life immune challenges. And in our mouse model, we saw the same delayed puberty onset. We saw also fewer eggs in the ovary. And because it's a mouse, we could get into the tissues and found a particular gene which encodes for a protein, which was responsible for both the altered function in the brain and the altered function in the ovaries. We also looked at the DNA that encodes this gene and found it had more of these methylation markers, these epigenetic markers, which are responsible for the lower levels of expression of this particular protein. And what was really exciting, when we looked at the same gene in the human DNA samples, we found the same methylation marks. So in this study, I think it shows really nicely how we could take very limited data which is available from the human populations and combine it with our mouse model and molecular studies to understand the basis for both the later puberty onset and the earlier menopause, which are responsible for the shorter reproductive lifespan in response to the more harsh early environment. Switching gear a little bit, we've been trying to consider the ways in which we can utilize the information or the knowledge that we're generating from how the epigenome plays a role in regulating reproductive function. And specifically in this example, we were looking to see how we could shut down reproductive function by blocking the expression of these pituitary gland hormones, LH and FSH. And we considered that we could do that by utilizing the brain hormone, GnRH, to take to the cells which produce these hormones an effector molecule, which would shut down the gene expression. I'll get to the effector molecule in just a minute. But the concept here is that the GnRH hormone is attached to a packaging element which carries the DNA into the cell. So the GnRH binds its receptor, which is only found on these cells. The packaging element in the DNA detaches and goes into the cell, and the DNA is then expressed as a protein. So this protein is a protein that we designed. We designed it to contain two components, one of which recognizes and binds very specifically to the gene that we want to shut down. The other element brings in other proteins, which at the very specific site cause the DNA to close up. This is utilizing the epigenetic mechanisms. What's really neat about this system is that after the DNA is no longer present, the DNA that was delivered, the protein that was expressed is no longer present. The epigenetic packaging is recognized by the cell, which tries to keep it closed. So what we're developing here is it's a form of gene therapy without all the complications of gene editing, whereby we can target a specific gene and get the epigenetic mechanism to close down the expression of the hormone, and it should stay shut down for long term. We initially started this project with the aim of blocking reproductive function to control animal populations, but this certainly has potential applications also for human health when we need to shut down the production of steroids. For example, in certain types of cancer, in polycystic ovary syndrome, in endometriosis. But we can also utilize exactly the same system with a slight tweak in the DNA to shut down the other pituitary hormone, FSH. And this is important because the FSH levels got very, very high at the time of menopause. And they've been attributed with some of the effects of onset of Alzheimer's, osteoporosis, and metabolic complications that are exacerbated at the time of the menopause. But what we really have here is a very versatile platform where we can change the DNA to encode whatever proteins we want, and we can change the targeting protein to target different populations of cells. So we think this is a very exciting platform which may have multiple applications. We still need to take it a little bit further. Before I leave you, I just want to credit the people who've done the work. My former PhD student, Ben, did the work on the mouse that I told you about earlier. And Lilach is my research assistant who did the development of this technology to deliver specific DNA into these particular populations of cells. So thank you for your attention. And I'd like to pass now to my colleague, Nabieh, who's also from the Faculty of Biology. And Nabieh, please.
Hi, thank you. Hi, hello everybody. I'm very excited to be here. And I would like to tell you a little bit about what we are doing here in my lab at the Technion. So, as you can tell from the title, I work on DNA damage repair and cancer, and I will convince you in a minute that these two fields of research are very tightly linked to each other. So in each cell in our body, we have around 2 meters of DNA. And these 2 meters are folded and wrapped in a very clever and sophisticated manner inside a very tiny nucleus. And this folding process of the DNA into the nucleus is really well regulated. And just to give you an idea how difficult it is, it's like taking the Eiffel Tower in Paris, which is around 330 meters long, and putting it inside a single sesame seed. And this wrapped and condensed DNA contain all the genetic materials that we need in order to build our body and maintain it. And it's actually quite shocking to know that our DNA is under continuous attack. And for example, each single cell in our body could experience hundreds of thousands of DNA lesion, or what we call it mutation, every single day. As you can see here, more than 50% of the DNA lesion are single-strand break, where we have a break in one strand while the other remain intact. And this break can be easily repaired using the intact template here, the intact strand as a template for repairing the broken one. On the other hand, the double-strand break, where we have a break in both strands of the DNA, is considered the most cytotoxic form of DNA lesion. And a single unrepaired double-strand break can lead to cell death and cancer, as we will see in a minute. Luckily, in our cells, we have hundreds of proteins that are involved in the repair of the different type of DNA lesion that we encounter every day. The problem starts when when one of the DNA repair protein is missing. Let's say that we have a mutation on one of the DNA repair protein that is required for repairing double-strand break, leading to defective double-strand break repair, and this leads to accumulation of mutation, genomic instability, and we can end up with cancer. And the example that I like to give here is a mutation in BRCA1 gene. A mutation in this gene, which is cellular function, actually is to repair double-strand break. When this gene is missing, there's no repair of double-strand break, and this can lead to cancer, such as mainly breast and ovarian cancer. And actually this example highlights that DNA repair and carcinogenesis are tightly linked to each other. So what we do in our lab, we basically induce DNA damage in living cells. We use several methodologies to induce DNA damage. One of these assay is called laser microirradiation, where we can induce DNA damage at a specific region within the nucleus of living cells. And then we capture the protein that are recruited to the site of DNA damage. And our assumption is that proteins that are recruited to the site of damage are somehow involved in the repair of the lesion that we induced. For example, here you can see the recruitment of a protein to the damaged area. And after we identify this protein, we use several methodologies to understand what exactly they are doing at the site of damage. And during the last year, we are putting a lot of effort trying to translate our discovery into diagnostic and therapeutic approaches to selectively kill cancer cells. And today I decided to give you an idea about a story. It's yet unpublished. Hopefully it will be submitted in the next 2 weeks. It's focused on a gene called RBM10. So we identify a novel function of this gene in DNA repair. But what is exciting about this gene that it's mutated in 21% of lung cancer patients. And our goal is to selectively kill lung cancer cells that have a mutation in RBM10. So how can we achieve that? I'll show you in the next slide the concept that we are using in order to selectively kill cancer cells. So let's say that we have here two cells, normal cells that have two hands or two genes. One is RBM10 and the other one, let's call it gene X. And the second cell is a cancer cell that had a mutation in RBM10. And the mutation in RBM10 caused or convert the cell from normal cell to be a cancer cell, okay? So let's say that now we can inhibit using small molecule inhibitor, we can pharmacologically inhibit gene X. Let's see what will happen to the normal cell. So if we inhibit gene X, then we will end up with a cell that had only one hand, which is the RBM10 hand, and therefore the cell can survive, okay? But if we do the same for the cancer cell and now we inhibit gene X, then we will end up with a cell that lost two hands. The first hand is lost initially because we have a cancer cell and there is no RBM10. And then we inhibited gene X and we end up with a cell with no hand and this cell will die. So we use this concept called synthetic lethality in order to selectively kill RBM10 mutated cells. The thing, what I didn't tell you, that gene X is around 25,000 genes. So in each cell, we have around 25,000 genes, and our mission was to identify which gene out of the 25,000 genes, if we knock it out or if we delete it, it will selectively kill only cells that have mutation in RBM10. And to do this, we use a technology, a powerful technology called CRISPR-Cas9, where we can knock out each of the 25,000 genes. And then we will ask which one, if we knock it out, it will kill only the cancer cells that have mutation in RBM10, but has no detectable effect on the normal cells. We did that screen and we identified around 221 genes. One of these 221 genes called WE1. So to test ourselves, test if the screen is good, what we did, we generated lung tumors, human lung tumors, and using mouse model. If you can look here at the third row, these tumors derived from RBM10 deficient cells that have a mutation in RBM10, resembling the 21% of lung cancer patients. And now we treated them with a small molecule inhibitor that inhibit a gene called WE1 that we identified in our screen. And you can see very nicely how the tumors significantly shrank, okay? And the size was reduced. And we are very excited about this observation because it provides the preclinical basis for testing our small molecules on samples taken from lung cancer patients. So this is more or less what I wanted to say today. And I hope that I managed to convince you that like our strategy is pretty much very promising strategy where we can combine basic science with and trying to translate it in order to establish new therapeutic approaches for targeting, selectively targeting cancer cells. Thank you.
Thank you so much, Noam, Philippa, and Nabieh. Viewers, please give us one moment while we bring all of our speakers back to the screen. This was a fascinating discussion, and I look forward to hearing more about it. It's now my pleasure to welcome Joey Selesny, the ATS Regional Director of Development based in Bloomfield Hills, Michigan, who will moderate the Q&A for us. Joey?
Hi, thank you for having me. I'm so happy to be part of this program. So we're going to start off with a quick question that was shared, and that is, what are some of the environmental effects that, this is for all three of you, what are some of the environmental effects that can take place that would affect some of your research? For example, where you live, or what you eat, or your exposure to the sun, all kinds of things that would change some of the effects.
So do you want to talk about exposure to the sun, maybe? Because I think it's in your arena.
I didn't think the question was for me.
The effect of exposure to the sun on DNA damage?
Yeah, so as I mentioned here, without being exposed to any exogenous agent, each cell could experience something like 100,000 of DNA lesion per day, and these lesions are spontaneous lesions, meaning that they arise during the metabolism of the cell. But the estimation is that each cell could experience much higher number of DNA lesion due to the exposure to sunlight like UV, smoking, and many other carcinogenic agents. I just missed the beginning of the question. I hope that I answered.
Yes, thank you very much. Moving on to the next question. If synapses constantly change, do we know how they create long-term memory? I understand that we forget and short-term things, but where do we store our long-term recollections?
I guess I'll take that one. I think this is the million-dollar question. I think that for a long time, the community was very excited about synaptic changes, and there was this kind of naive expectation that when you change the synapse, it just stays there forever. I think we're appreciating now, probably maybe a decade, maybe a little bit less, that the synapses change all the time. So first of all, it took a while to realize that there's a problem at all to start with. And once the problem is present, there have been several ways of addressing it. For example, some people suggest that perhaps we don't store a memory in one synapse, we store it in hundreds of thousands. And then if we average, the fluctuations, then they all average out, and what you're left is with something that's relatively reliable. Other suggestions, there were other suggestions, for example, that during sleep, somehow you retrain all the synapses that kind of got mixed up, and you reteach them what they lost during the day. I think these are good questions, but the true answer is we don't know. And in fact, when people now are looking at one level up, they're just looking at what the brain does. When you give it exactly the same stimulus over many weeks, it turns out that the brain is completely drifting. So now people call it representational drift. You give the same sound to the animal today, you give it 3 weeks from now, and the brain does something quite different. So it is a big question which we are trying to address, I think, in recent years, and I don't have a good answer for that.
Thank you very much. Noam, this question's for you. When you were talking about what the Technion Human Health Initiative does in terms of bringing collaboration within the different departments of the Technion, what about international collaborations? How do those work?
international collaborations are always something we're interested in. For example, I'm personally involved in one very big one with three groups in Germany, but 4 groups actually. But I think that we have a lot of work to do internally. For example, just to give you a very relevant illustration, 2 years ago, I didn't know Philippa. I mean, I knew she existed. I knew the name. We never talked. And now we talk all the time. Probably she doesn't want to talk to me anymore, but we communicate a lot. So I think, yes, international collaboration is something we are doing all the time. We've been doing it forever, but actually we have to collaborate, first of all, internally, because I think this is where we are missing.
Thank you. In terms of backing up to that environmental question, in terms of genetically modified things that we're eating all the time, also supplements that we can eat. I think this is a question for Philippa. With all the supplements that we could have to help us with our hormone balance and so on and so forth, do those things help, and should we continue to do those things?
Disclosure, my husband's in the business of organic chickens, so I need to be very careful what I say. We need to be very aware of what we eat is going to affect all of our biological systems. Certainly supplements, if you're talking about supplements of components of the diet that is lacking in our diet and we need, they're going to help. But I think there's probably in the market, there's a huge flooding of supplements, nutritional supplements, which are not under very careful regulatory control. And I would be very, very wary of them. I'm not saying that they're not necessarily required, but I would be very cautious of believing what I hear in the advertisements and the public press, because not necessarily what it says it is, and it's not necessarily that you need it, but you might need it. Obviously, the general practitioner here would be the person to ask if these supplements are really required.
Excellent. Another question for you. Can the insights about reproductive organs in women be used to develop new forms of birth control or even fertility?
So obviously, if we're looking for new forms of birth control and to improve fertility, we need the biological understanding. That's for sure. I think many of the birth control methods which are being developed now, or the emphasis in birth control technologies, is more non-hormonal ones. They're based more on interactions between the sperm cell and the egg cell. But there's definitely a requirement. Obviously, there's a requirement for a male contraceptive, and people are working on it. It's not something that's specifically under my radar at the moment. And the other question was about fertility. So there are so many causes of infertility or subfertility that we don't understand. In many cases, the PCOS, the polycystic ovary syndrome, and endometriosis, are major causes of subfertility, and they're really, really poorly understood. So I think there's a major drive and a major requirement to really understand the biological mechanisms that are causing these horrible diseases, which are incredibly common, unfortunately, and really causing fertility problems for a major part of our population. So yeah, this is where we need the money and the researchers and the scientific groups to really push forward a scientific understanding.
Excellent, thank you. One more question regarding to Nabieh, excuse me. When you're dealing with destroying, damaging proteins and recreating making that ability to damage them and then look for cures through that, you're focusing specifically on lung cancer. Is protein folding involved in all forms of cancer? Is that what creates cancer? Is the inability of the body to deal with that?
Yeah, it's a good question. Yes, I showed you here an example of RBM10, but we work on other proteins that are mutated in other type of cancer, like breast cancer. But also with RBM10, I just mentioned that it's mutated in 21% of invasive lung cancer patients, with a huge number of patients. But it's also mutated in many, many other type of cancer. like between 1 to 5%. Okay, I don't remember now the exact numbers, but it's also mutated in other type in cancer, and therefore inhibiting that small molecule inhibitor that we use, we believe that it can be applied also in other type of cancer other than lung cancer that have mutation in RBM10.
Can I jump in just for a second? The person who asked the question about protein folding, the whole issue of proteins folding, if anybody still remembers and hasn't forgotten my slide on the parts of the synapse, proteins are these really wonderful machines, but they're really tiny. And to understand them, we need to understand their structure. And part of what we're doing now at the THHI now is to create a very strong emphasis on developing the means and the personnel for helping our investigators uncover the structure of their proteins. It is a very challenging direction, but if anybody has followed the literature the last few months, things like machine learning and artificial intelligence have created huge breakthroughs in this. And so we're trying to get into that field too.
That brings me to another question. Recently, we saw that there was a partnership announced between Pfizer and the Technion, and I think with Shai Shinor's research. Is that an area that you all hope to be able to get to as well?
So, yes, indeed, there is a strategic partnership between Pfizer and the Technion, and indeed Shai Shinor was the leader of this initiative. And it's very deeply integrated into our efforts because AI is something that Technion is really a leader in. It's, I think, #1 in Europe in this arena. And there's a huge amount of work to be done on the interface between medicine and machine learning, and including structural biology, including drug development. And I think that's one of the reasons Pfizer is interested in collaborating with the Technion.
Okay, this is one last question that I have. How this, I think, for Professor Melamed, how far off is gene therapy that targets epigenetic mechanisms for cancer, endometriosis, from being realized?
I don't think it's that far off. It's definitely getting a lot of research attention. There's still more work to be done. Most of the problem in gene therapy is the targeting aspect, which is why we've been focusing on utilizing hormones which target very specific cell populations. And I think this is really the way to deliver epigenetic modifying drugs, which can have multiple different kinds of applications. How far off it is, it's going to be in the next few years. Obviously, the more money that gets funded into the right direction, it'll get done faster. But it's making good progress, and it'll definitely be within the next few years. But if you're looking at something like endometriosis, you need to know exactly what you need to target, what genes you need to shut down to stop all of this kind of inflammatory response. So the biology needs to know what we need to target, but the technology of being able to make those targeting platforms and deliver, for example, a DNA or a small molecule to a specific tissue or a specific cell type, that'll be within the next few years for sure.
Okay, this question has come up a bunch of times, and that is regarding something we touched on earlier, and that is which we talked about in terms of environmental effects. Is there anything that you can any of the three of you can recommend that one of our viewers or others could do to protect their bodies?
Eat well.
I would say mild exercise, definitely, and especially as we get older and we get sedentary. I can tell you a little anecdote as a cell biologist. It looks kind of trivial on the screen that you can keep cells on the microscope and look at them for weeks continuously. It turns out that we worked really hard and actually failed for a long time. We tried a zillion vitamins and amino acids and formulations. And at the end, the thing that really made the huge difference was just replacing the media slowly, just like it happens in the brain. And if anybody does exercising or a brisk walk, he's probably going to have more circulation in his brain, and it's probably going to do good. That's as far as I risk my humble knowledge of cell biology to extrapolate. But this has been like a striking observation beyond anything we've ever seen.
Well, thank you very much for all three of you giving us such wonderful answers. There are lots of questions that we were not able to get to today that hopefully we'll be able to respond in follow-up webinars or in a follow-up email after this webinar is over. So I just want to thank you for your wonderful conversation. And Janie, back to you.
Thanks, Joey. My sincere thanks to all three of our panelists, Noam, Philippa, and Nabieh, who have described so clearly why Technion Professor Uri Savan's vision, which created the Technion Human Health Initiative, is so vitally important. This transformative endeavor represents the most significant focused investment in the Technion's history. It's critical to its future and to that of the State of Israel. No other university in the country, and only a few around the world, are so well positioned for this undertaking. The Technion already embodies a multidisciplinary atmosphere and culture of collaboration, yet lacks the resources and infrastructure for a project of this size and cost. As Israel turns 75 years old this year, and the Technion celebrates its centennial next year, now is the time for all of us to help this vision become a reality. If you'd like to find out how to support this amazing initiative, please reach out to your local ATS representative or send an email to ats.org. And thank you to all of you for joining us today. This program has been recorded, and you'll receive a link to that in the next few days. In the meanwhile, enjoy the rest of your afternoon, and bye-bye.